Welcome back to my iBio seminar series part 2. Again, I'm Xiaowei Zhuang. I'm a professor in the Department of Chemistry and Chemical Biology and the Department of Physics at Harvard University, and an investigator of the Howard Hughes Medical Institute. Now, in part 1, I introduced super resolution fluorescence microscopy, and in particular, the single molecule based super resolution imaging method, STORM. And in this part, I will give you a couple of example applications of STORM in microbiology and neurobiology. So I will upfront say that in this application, we use photo-switchable fluorescent proteins to do the STORM imaging. And in this application, we use photo-switchable dyes to perform STORM imaging. So first, our application in microbiology. And one areas of application that I'm very excited about is bacterial imaging. Now, bacteria, these are tiny cells, and if you look at their width, the width is just several hundred nanometers, or sometimes approaching a micron or so, and that is nothing but just a few times of the diffraction limited resolution size. So because of these cells are so small, it has already actually been traditionally difficult to image them. And because of this difficulty in imaging, our understanding of the cell biology of bacteria actually lags far behind our understanding of the cell biology of eukaryotic cells, for example, mammalian cells, which is quite funny and ironic considering that these are arguably simpler cells. So super resolution fluorescence microscopy offers an ideal approach to solve this bacterial imaging problem. So here I'm just going to give you one example where we study the nucleoid and chromosome organization of E. coli using STORM in collaboration with my colleague Sunny Shi at Harvard. So we know that bacteria is small, and its DNA structure inside the bacteria is even smaller. In fact, if you spread out the DNA of an E. coli cell completely, the contour length is almost 1 millimeter, but it has to be compact into a much smaller structure, a submicron structure called the nucleoid. And this compaction is facilitated by a number of different proteins in the bacteria, among which are these major nucleoid-associated proteins. In E. coli, I listed 5 of them here: HU, HNS, IHF, FIS, and STPA. And we imaged their distribution using STORM. And here, for example, what you see is actually a live cell STORM image of HU in E. coli cell. And you can see that this HU molecule pretty much are scattered throughout the entire nucleoid. And here, shown on the left, is the bright field image of the E. coli cells. And this phenomena is also found to be true for these other NAP or nucleoid-associated proteins like FIS, IHF, and STPA. In contrast, HNS distribution looks drastically different. Now, instead of being spread out throughout the nucleoid, they form these tight clusters inside the cell. And as you can see, that these clusters are much better resolved in the super resolution STORM image as compared to the conventional image. These clusters are quite static, or they don't move around that much. You know, here what you see is actually a live cell movie of HNS. It almost looked like a still image because they're not really moving around that much. And by quantifying these type of clustered structures in many cells, and we found that these HNS clusters are organized in a very regular way in two different senses. One is you can see the number of clusters inside the cell. That actually increases in general with the length of the cell. The longer the cell, you see more clusters. And for the newly dividing cell, under conditions that we know that there's only one copy of chromosome inside the cell, we found basically 2 such clusters. For example, here in this image, and then you can see here in this bar graph. And that's one way of regulated organization. And the other one is their position. You know, I won't belabor you with these distribution histograms, but just to tell you that by quantitative characterization of many such cells, that we found that these 2 clusters tend to be positioned along the long axis of the cell at the quarter cell positions. And what's the mechanism that they cluster? To investigate that, we did mutation studies. So HNS molecules is known to have this type of structure where it has 2 domains, a DNA binding domain near the C-terminus and a dimerization domain at the N-terminus. So to see how they affect the clustering, what we did is we used this previously established mutation, L30P, to actually disrupt the dimerization tendency of the molecule. And then once we introduce this mutation into endogenously expressed proteins by chromosome insertion, you can see that the clusters are no longer there and the molecules are now again scattered throughout the nucleate, just like HU. And then when we use this previously established mutation that actually disrupted DNA binding, we found much fewer localizations and telling us that these localizations we see are indeed due to DNA-bound molecules. So this tells us that the cluster formation is due to the interaction, the self-interaction between the N-terminal domain of the molecules that are bound to DNA. Now, functionally, HNS is a global transcriptional silencer. It represses about 5% of all E. coli genes, a total of around 200 of them. So to see whether the cluster, how they're related with the repressed genes, we actually looked at 2-color imaging of HNS clusters and the gene locus, and specifically 3 of them, the HDEA gene locus and the HCHA gene locus. And these are genes that are known to be, their expression are repressed by HNS. And as we also show here with RT-PCR, that indeed their expression level is substantially reduced in wild-type cell as compared to HNS deleted cell. And as a control, we looked at the LAC-Z gene locus, and LAC-Z, you can see its expression level is not repressed by HNS. And then here is the 2-color super resolution images of HNS in purple and the gene locus in green. And as you can see on this left 2 panels, there's substantial co-localization between the gene locus and the HNS cluster, while there's no such substantial co-localization for this control gene. Actually, by sequestering these gene locus into the cluster, We also found that the gene locus has been moved by substantial distances inside the nucleoid by these clusters, as demonstrated in these distribution maps where we compared the distribution of the gene locus in wild-type cells and in cells where HNS is deleted. And here, when you see 2 lobes and 4 lobes, that doesn't mean there are 4 such gene locus inside the cell. They just mean these are the positions where the gene locus are most likely to be. And you can see that the most likely position has been moved substantially by the HNS clustering effect in both of these 2 cases, while in the control genes you can see that the most likely positions are not moved much when you delete HNS. And as I said, since HNS is a global transcriptional silencer which represses about 200 genes, and they actually have even more than 200 binding sites that are distributed throughout the E. coli genome broadly. So by sequestering these sites into the clusters, we think that HNS has a global organization and compaction or condensation effect on the bacteria or E. coli chromosome. To see that these genes are indeed brought into proximity in a more high throughput way, we utilize this technology or this method called chromosome conformation capture developed by Nancy Kleckner and Job Decker a number of years ago at Harvard. And in this technology, the gene locus or the DNA locus that are brought into proximity by proteins are probed by cross-linking and then later on examined by PCR, for example. So we use this to see, among these locus that are regulated, or the genes that are regulated by HNS, whether they are indeed brought into proximity. So here in this plot, what you see, the black circles are the genes that are regulated by HNS. And as a comparison, we also put in these control loci by the red crosses. And then we probed the pairwise proximity by their cross-linking frequency, or cross-linking efficiency. And as you can see here on this bar graph, among the control pairs, between the negative control size or between the control size and the HNS regulated site, these control pairs give you very low cross-linking frequency, while in contrast, the pairs that are formed between 2 HNS regulated loci give you much higher cross-linking efficiency. And moreover, this cross-linking frequency is dependent on HNS, because if we delete HNS in the cell, you can see the cross-linking frequency drop down to the control level. So with this, we have this model picture of how HNS proteins could help organize chromosome inside the cell. And that is, they form tight clusters in the nucleoid, and quantitatively from each copy of chromosomes, there are about 2 such clusters, and they form by oligomerization of the DNA-bound HNS through their N-terminal domain interaction. And by sequestering many gene locus or DNA locus broadly distributed on the chromosome into these tight clusters, we think that this molecule helps DNA folding and condensation by serving as organization centers that anchor and juxtapose numerous of a DNA HNS binding sites on the bacterial chromosome. So that's the first example. And the second example, as I mentioned earlier, is in neurobiology. And in particular, in this example, we are investigating the sub-synapse structures, or the molecular architecture of synapses, together with my colleague Catherine Dulac at Harvard University. As we know that synapse structures are the fundamental functional unit inside the brain that connects neurons and their target cells. And I like to say that synapse is also an ideal subject to investigate with super-resolution fluorescence microscopy. And the reason is it's also tiny. The size of a synapse is, say, several hundred nanometers. Again, not much larger than the diffraction-limited resolution of conventional light microscopy. Inside this tiny structure resides hundreds of different types of proteins. Some are existing in high copy number, and therefore you are seeing actually thousands or more molecules densely packed in this basic functional unit in the brain, that all these molecules are working in a concerted manner to mediate the neurotransmitter mediated signal transmission between neurons and their target cells. So it is thus essential that we understand how these molecules are organized and how do they interact with each other in order to understand the functional mechanism of this structure. And this is very difficult to investigate with a conventional light microscopy tool, as I illustrate in this image, this conventional image of synapse structures that are labeled with two proteins. One is the presynaptic scaffolding protein bassoon in red, and the other is the postsynaptic scaffolding protein HOMER1. And as you can see in this image, there's severe overlap between these two structures, and also in places multiple synapses are overlapping with each other. And here is the storm image of the same field of view, and then you can move, let me move this way so that you can see the storm image more clearly. And then if I zoom into a smaller region, you can even more clearly appreciate the difference between the conventional and storm image, where these two synapses are mistaken as one in the conventional image. Here we resolve it very clearly with the storm image. And then also you can see the clear separation between the pre- and postsynaptic scaffolding proteins. Now, because this is a three-dimensional imaging method, you can actually see the separation of these two, right here, of these two structures even more clearly if I show you this movie that rotates and then shows you all 3D perspective of this structure. And then you can see that at the cross-section view, you can see the clear separation of the presynaptic scaffolding protein and postsynaptic scaffolding protein separated by the synaptic cleft. And here is the en face view showing you how they are distributed laterally. And we can do three-color storm imaging. And here, adding the third color and actually add a substantial capability for us to determine the molecular architecture of the synapse. So here is our experimental design. We use two such markers, again, bassoon and Homer, to set the reference frame. And then we systematically swap out the third marker. So for example, in this image, what you see is GLUR1, which is a subunit of the AMPA receptor, and as expected, it is distributed in the synaptic cleft. And then if we keep changing this third marker, and then we can actually determine the position of many different synaptic proteins in this framework set up by Bassoon and Homer. And then here is, using this approach, we determined the position of 10 or so synaptic proteins along the transsynaptic axis like this direction. And here you can see that such an architectural map really gives you very rich information about how proteins are distributed and laid out in the synapses. And I won't go into explaining the implication of each individual one due to time's sake. And we can not only determine the axial position of these molecules along the transsynaptic axis, we can also, because this is a 3D imaging approach, to have an en face view to let us to determine this lateral distribution of molecules along the synaptic cleft. And for example, we looked into the lateral distribution of neurotransmitter receptors, which are of particular interest because they could affect the transmission property of synapses. And then here, what you see is the distribution of GLUR1, which is an AMPA receptor subunit, and NR2B, which is an NMDA receptor subunit, with respect to the postsynaptic density marker HOMR1. And we can see both central synaptic distribution, where the receptors are almost exactly overlapping with the postsynaptic density, and a perisynaptic distribution, where the receptor forms an annular ring around the postsynaptic density. Now we can not only get this kind of a lateral distribution, we can also get quantitative information on the composition of receptors in the synapses, which is another important property that could determine the transmission property of synapses. Now if you look at these two images, you might even think of these as two-color images, because you can see green and blue. But in fact, these are actually three-color images where all three probes, HOMR1, GLUR1, and NR2B, are simultaneously probed. There's only largely two colors because this protein, NR2B, is largely missing in these synapses. In the same brain area, we can also see distributions or composition like this, where you can see a large amount of red signal indicating actually a lot of NR2B subunits are located in this synapses. So it tells you that the composition of synapses can be highly heterogeneous even in the same brain area. By quantifying the neurotransmitter receptor composition over many synapses in a couple of brain regions, we actually come up with a pretty surprising discovery, and that is we found that in this adult accessory olfactory bulb, which is short named here as AOB, we found a large population of synapses that predominantly has NR2B, but have very few GLUR1. And if we further look at the postsynaptic density marker, HOMR1 and other postsynaptic density proteins, we found that these synapses that predominantly has this NMDA receptor subunit NR2B, but lack largely the AMPA receptor subunit GLUR1, they also have a very small amount of the postsynaptic density protein. So all these put together, these three signatures, low AMPA receptor content, high NMDA receptor content, and low postsynaptic density content, could mean that these are likely immature synapses or silent synapses. And I say this is a surprising discovery because this is found in adult mice in their accessory olfactory bulb or AOB regions. And while these immature or silent synapses has known to be present in large number in the early developmental stage, and they're known to be over the time mature and disappear or get converted into mature synapses before the animal reached adulthood. And indeed, in the MLB region, the main olfactory bulb region, you see very few of such synapses. So to see whether these potentially immature or silent synapses could be converted into mature ones by stimulation, we utilized this wonderful technology, optogenetics technology pioneered by Carl Dijkstra's lab at Stanford University. So here we took advantage of a mouse line that is expressing channel rhodopsin in the verminasal organ, sensory neurons, which synapse with mitral cells in the associated accessory olfactory bulb. And this mouse line is generated by my colleague Vanke Murphy's lab. And then we use light to stimulate these AOB synapses. And we want to see after stimulation whether these synapses could mature into a more mature synapses. And then here what you see is the blue distribution is before the light stimulation, and the green distribution is after stimulation. You can see indeed these synapses that have low GLUR1 content indeed matured towards the site by accumulating GLUR1 and also by decreasing their NR2B content. So with this example, I hope to have shown you that 3D Storm allows quantitative characterization of molecular architecture of cellular structures as small as synapses. And of course, it can also be applied to many other tiny cellular structures that are difficult to study by conventional approaches. And indeed, we have already made discovery with this kind of super resolution approaches. So with that, let me again acknowledge the people who has done the work. And the bacterial imaging work is done by a graduate student in the lab, Wenqing Wang, as I said, in collaboration with my colleague Sunny Xi's lab and his graduate students Junwei Li and Chongyi Chen. And the synapse work is done by Bo Huang when he was in the lab in collaboration with my colleague Catherine Dulac's laboratory and his former postdoc Ada Shtani. And this concludes the second part of my iBio seminar series. And then the third part will be on single molecule FRET study of a couple of biological systems that are involved in gene replication and expression and regulation and so on. Thank you.